or Twitch and look for the Radio Artifact channel, and you can find our presentations there. So this evening we're back talking about the environment and our fragile planet. Our speaker this evening is Valerie Pence. She's the Director of Planet Research at the Cincinnati Zoo Crew. Valerie is going to talk about, as her title says, Saving All Planets, the Challenge of Planet Extinction. She's going to talk about, globally, plants being under threat and what is being done to try to save plants, especially some of the specific work that she is spearheading at the Cincinnati Zoo. So please welcome Valerie Pence.
Well, thank you, Bob. Can you all hear me? I guess I got to be real close to this. But I'll try to, maybe I better try this. Is that good? Okay. Nobody has ever accused me of speaking too loudly, so I know I need this. Okay, well, thank you so much for inviting me to be here. This is a real exciting thing for me. I haven't been to one of these before, and I think now I'm going to be trying to be here or tune in because it looks like a great program. What I want to do is, as Bob said, talk about plants, and that's what we do. Excuse me, I swallowed something right before I ended. Something wrong here. At the Cincinnati Zoo and Botanical Garden. And I work at the Center for Conservation and Research of Endangered Wildlife, which is called CREW. And I think you've had a speaker from CREW before. Lindsay Van Sant said she had spoken to you all and had a great experience. She works with our CAT program there. I direct the plant research program, and so our work is all plants all the time. So that's what I'm going to be trying to talk to you about tonight. I'm sorry. I'll start out by showing you this picture. I just said we're going to be all plants, so I sort of gave it away. Because I was going to ask you what you saw in this picture. There is a chicken, right? And I think most people's eye goes to that naturally as you look at the picture to start with. But there's a lot more in that picture as well. And there's a lot of plants. And I think most people would say, yes, there are plants in there. But there's a lot of different plants. And I'm not going to try to identify them because I can't. But, there's sort of feathery plants. I won't try to point them out. But in the left-hand corner, there's some feathery plants. There's some white flowers at the bottom. There's some grass in the back. There's lots of different stuff in there. There's a bush. There's herbaceous plants. What I'm going to be talking about tonight are plant species. And thinking about plants not just as background, not just as the environment or the setting for our lives. They are that, definitely. But the plant world is made up of species just the way animals are. And so at CREW, we're very interested in the species as well as just the plants. So there's another picture that I found that I thought was sort of nice. Of course, you see the bison in there to start with, but there's a lot of plants around it. And it's not just, it's not just the green background. There's trees of certain, species, there's grass, there's herbs, there's just all sorts of stuff in there. If you go out into your lawn, unless you have a perfect lawn, there's probably more than one species there as well, if you look closely. So it's really quite interesting. And just to tell you a little bit about how geeky I am about plants, when I watch a movie or something on TV, and I try not to tell people or say too much about this while we're actually watching because people get annoyed at it. But, look at this picture from the African Queen. What do you see? There's a lot of plants there. And even if you don't know too much about plants, you probably can see there's a palm tree in there, as well as other tropical trees. There's Humphrey Bogart in the I don't know what that was, a swamp, a lake, something, with lots of reeds around him. Those are species. There's some algae growing on the water surface. I know there were leeches in there too, which was really gross. But there's another scene from the African Queen. A lot of little Fionas coming at them. But look at the side of the river. There's bushes, there's trees. There's a few trees that actually pop up really tall. You know, those are all different species. And then this is actually from the Indiana Jones Raiders of the Lost Ark. You know, I see something like that and I say, what vine is that? You know, that tree on the left has really big leaves, it looks like. And then there's other things in there. So it's fun to sort of think about those kind of plants. And if you're really good, I'm not really good at identifying plants, particularly tropical plants, but some people who really are can watch movies and tell you when the directors put in the wrong plants. They just got a house plant and stuck it in there. And I've heard that people who are really good at bird calls can do the same thing. That there's a lot of bird background music in movies that's really wrong. But what I'd like to talk to you about tonight and give you 3 take-home messages on are these: why plant species are important, and some of the global work that's going on to actually save plant species, and then sort of focus down at what we do at Crew to work on plants. So I think if you thought about it, you probably know a fair amount about plants in general, even if you're not a botanist. We know that plants, they provide us with a lot of things that are really important, like oxygen, and most of our food comes from plants, either directly or indirectly. Plants can photosynthesize and take the light from the sun and combine it with carbon dioxide and make all the sugars that are needed for itself, as well as for all the animals and humans on the planet. So they give us oxygen, they give us food, and these days we're learning a lot about carbon sequestration, which is what they do, taking up carbon dioxide and then converting it into food. So that's become even more important in these days of climate change. So I think it's pretty safe to say that all life depends on plants. But that, and I'm not trying to say anything bad about plants in general. It's good that we know that we have plants out there and that they really are they are the life system of the planet. But what do we know about plant species? As I showed in some of those pictures, plants, you know, if you look closely, even if you don't know what the species are, you can say, this is a species different from this one. Plants are different from each other. And because they're different from each other physically, they also do different things chemically. So they produce different kinds of chemicals. And we do different things with those chemicals based on what species they're coming from. So the fact that there is this multiplicity of species out there is really, really important to our lives. If you just think about maybe what's in your kitchen. The kitchen itself may be based on a lot of plants. You might have oak cabinets or floors, cotton towels. There's oranges on the counter, a teapot, lots of things that we eat and use in cooking that all basically come from plants. Our medicines are based heavily on plants. Even if they're made synthetically these days, many of them are based on chemicals that were originally discovered in plants, and they've just been maybe modified slightly in the development of the pharmaceuticals. Aspirin, of course, comes from the willow bark and has been modified slightly, but has been one of the major pain relievers for many, many years. And they're Taxol, anti-cancer drugs, a variety of those things. So there's a wealth of these kind of chemicals that are made from plants, and plants usually make these because they are somewhat defensive chemicals for them. There's chemicals that are made to defend against insects or herbivores, those kinds of things. But we've been able to use them for our benefit. It's been said, though, that there's probably only about 10% of the plants out there, the plant species out there, that have been evaluated for even one use for humans. And those kind of uses might be not just medicines, but fibers and foods, and just all sorts of things that people get from plants. There's just so much out there that we really don't know yet. And that's one of the driving forces behind trying to preserve the plants that we have, because we just really don't know what we're giving up. We don't want to get rid of them before we know what they actually are. Another way I like to think about species is that every species has, I like to say, every species has a home. Every plant has a home somewhere. Every plant that you see is native somewhere on the earth. You might think of them as weeds, like the dandelions, or very invasive plants, like the Amur honeysuckle. But those actually are from some other place on earth. They were native somewhere, and then were brought here. Hostas, for example, came from China. They're not invasive, they're just beautiful, and perfect deer food, at least in my yard. But they came from somewhere, and they're native somewhere. The ginkgo tree is planted widely as a beautiful street tree. It was native to China, was almost extinct, and then was brought in as a very important horticultural tree. Dandelions, by the way, came from Eurasia, and were brought here obviously by the Europeans who came over, and the Amur honeysuckle was brought in a couple times sort of ornamentally in the early 1900s, and became quite invasive. But there was a professor over at NKU a number of years ago who was studying the honeysuckle, and he went to China to look at it in its native habitat. And I saw the pictures. I wish I had one with me here, but he had a picture of it, and it just looked like this, sort of straggly bush in the forest by itself, that was just alone. And here it grows in all these masses of bushes, so it had a different kind of environment there than it does here. Obviously, it probably had some predators or perhaps some diseases that kept it at bay there that it doesn't here, but it's become quite invasive in our area. I also wanted to just share a few fun facts and maybe some not so fun facts about plants. There are actually about 305,000 species of plants globally. That's compared to about 5,000 mammal species globally. You may have heard that there were maybe 350,000 or 400,000 species of plants. Recently, there have been efforts to create an actual list of all the plant species in the world. And people have found, that some of the species had 2 names or 3 names or 4 names because somebody named it in England and somebody else named it in France or whatever. So they've really narrowed it down and identified each plant as a single individual and gotten all the duplicates out of there. So right now it's about 305,000, but they are actually discovering new species even now. And so the number will probably grow. If you look at the number of species in different countries, it's sort of interesting. In the UK, for example, there's about 1,400 native species there. In the US, we have about 17,000. We're a much bigger country than the UK. That's part of it. But if you look at China, they have 30,000 species that are native there, and 40% of those are only found in China. They're endemic, as we'd say. 51,000 species in Colombia, which is not that big a country geographically. And a third of those species are only found in Colombia. So there's some areas of the world where there's an incredible richness of species, particularly in the tropics. And those are areas that are also under great threat by habitat loss. So that's one of the concerns that people have right now for plant species. The latest evaluation has said that 2 out of every 5 plants is threatened with extinction at this point. So that's about 40% of all the plants on the planet. And the rates of plant extinction right now are about 50 to 500 times the sort of background extinction rate. We know that plants and animals go extinct just naturally over time, but they've estimated that the rates are much, much higher right now. Another sort of interesting fact is that more than half the species on the Federally Endangered Federally Endangered Species Act list right now in the United States are plants. But plants receive only about 5% of the federal funding for endangered species, so quite underrepresented in the funding area for preserving the plants. Well, I mentioned that habitat loss is a major threat to plant species. That probably is the greatest threat right now. And habitat loss primarily to agriculture and human development of cities and roads and such. Unsustainable harvesting is also a problem with certain species, particularly certain kinds of timber species. And then climate change is becoming more and more of an issue and probably will become a greater threat in the future. When you think of plants that grow on mountains, for example, as the climate warms, they migrate up the mountain, essentially. And when you get to the top of the mountain, there's no other place to go. And so those are some of the concerns that people have right now for plants. Well, what's being done to preserve plant species? We know that conservation is the term we use to indicate work that's being done to preserve plants and animals. And just to review our Latin terms here, in situ conservation and ex situ conservation. In situ conservation means on-site conservation, that is, conservation of plants in their natural habitat. Ex situ conservation is what we do. It's off-site conservation, meaning we're preserving the plants in an area not in their native habitat. And so I have a question for you. What do you think, what portion of the world's land area is preserved right now? Would you say 70%, 30%, 12%, or 2%? Any guesses? 2? 3? Pardon? 3? Well, it's actually 12 globally. And if you look at a list of the countries by their percentage of land of their country that's preserved, and this is not the whole list, there's 192 countries on the list. I picked out some of them in order. Venezuela has 56% of its land preserved, and the United States is 119th on the list. Out of 192, we have 13%, so we're a little bit above the average. But a recent study came out that suggests that we need globally about 44% of our land set aside to preserve the world's plants and animals. So I know that there are efforts that are beginning to take shape to try to promote that in countries around the world to increase the amount of preserves that are there. Excuse me. Well, I'm going to talk a little bit now about ex situ conservation, off-site conservation, which is what I'm most familiar with, and start with agricultural species. We work primarily with wild species, but there's a great effort around the world to preserve agricultural species because our lives basically depend on that. It's an effort to maintain the food security of the world. And there's an organization called the CGIAR, which has a really long name. I think it's the Consultative Group for International Agricultural Research. And it's a global network of institutes around the world that focus on different areas of agriculture. And I don't know if you probably can't read that real well, but in Mexico, there's one that focuses on maize and wheat, corn and wheat. In Peru, there's one on potatoes. In Africa, there's one on African rice. Southeast Asia has a forestry institute, which deals with a lot of tropical forestry. And in the Philippines, there's the International Rice Research Institute. There's a number of other ones on there that deal with other more groups of plants, dryland plants, tropical agricultural species. And what these groups are trying to do is preserve all the different lines of particular crops. And when I say line, I mean plants, a variety that is adapted to growing in a certain area under certain conditions. Because rice that grows in certain areas of India probably does not have the same adaptations as rice that's growing in Louisiana or in Japan. So what they're trying to do is collect all those as a bank, an insurance policy for the food security of the future. And I'll just pick one of those institutes. This is the International Rice Research Institute in the Philippines, just an aerial view of them. And I love that picture because I never realized rice came in all those different colors, but it does. And it's so important to maintain those lines of the crops because each line holds genetic information that could be useful in the future, particularly as climate changes and areas change. There might be a line in there from one place that now is going to grow well in another place. So it's very important. And this looks into their seed bank of the Rice Research Institute. They have over 130 accessions of rice, and you can see there they have their jackets on because it's very cold in these seed banks, and I'll talk a little bit about that in a minute. But you can just see all of the bags and containers of rice samples that they have. Here in the United States, we have a whole network as well of seed banks and germplasm banks of various kinds. The USDA runs these, and they are all over the country, and they focus on different things. There's one actually in Columbus, Ohio, at OSU, that is the Ornamental Plant Germplasm Center, and they do a lot of research on ornamental plants and have a seed bank there for those species. But the sort of the central seed bank for crop species in the United States is the National Laboratory for Genetic Resources Preservation, or the NLGRP, and they are located in Fort Collins, Colorado, and have a major facility there. And this just sort of looks into it a little bit. You can see on the upper left-hand corner another person in a jacket walking through the seed bank there. And on the right, you can see that they also keep a lot of their material in liquid nitrogen. And I'm going to be talking about that a little bit more in a few minutes. But those large metal tanks, they're really big, like that big, are just full of samples, hundreds, thousands of samples of corn and wheat and various things in liquid nitrogen. And you can also see that they do a lot of research. Those other pictures represent their germination studies, what they do. I mean, if you put something in a seed bank, you need to also make sure it's not dying in there. So they take out samples every few years and test them. And that way they can learn how long things will last in the seed banks, because seed banks haven't been around all that long. And so it's a big research project as well. And if the viability of those samples is dropping, they'll take them out and grow them up and collect the seeds again and rebank them. But that's a lot of work. And so you want methods that are going to keep those seeds for a very long time. This diagram shows a little bit more about seed banking. And it sort of divides seed banking up into 4 basic steps. If you look at the diagram at the top of it just represents the plants that you collect seeds from. So if you're going to bank seeds, you've got to have a source of seeds. So you've got to have plants that are actually making enough seeds that you can take them and put them in the seed bank. The second step of seed banking is to prepare the seeds or process them for the storage. And what you have to do is remove the water from those seeds. Now you know that seeds, most seeds can dry down, most seeds that we are familiar with around here. They get very dry and blow around in the wind, or you go and buy a packet of seeds of tomato seeds or something like that, and they're very dry. And then you plant them and they rehydrate and germinate. But in the seed bank, they do this in a very systematic way. They put the seeds in a relative humidity of about 15% and dry them down to about less than 10% moisture content to make sure the water is gone. And why is that important? Well, water is your enemy when you're trying to store something in a freezer, and that's what the next step is. You, when you dry the seeds, and in a seed bank, you're going to put them into a freezer of about minus 20 degrees centigrade. And you know that if you take your fruits at home and you stick them in the freezer, when you pull them out, they don't really look quite the same. Excuse me. Because when water freezes in cell structures, water expands and forms crystals, and those crystals will break the cells and kill them. So in order to freeze something and keep it alive, you need to remove enough water so that you don't get that crystal formation. And that is the real basis of pretty much everything I'm going to talk about in terms of seed banking and the work we do, because you want to keep these tissues and seeds alive through the process. So the third step is you put them in the freezer and you leave them there for as long as you can. Now, I did mention that it's really good practice if you've got enough seeds to go in and take them out and regenerate them, a sample of them to make sure they're still alive. So the people are doing that when they have enough material. The final step is to take seeds out and recover them and germinate them. So you need to have seeds that are actually going to be able to do that. You don't want the seeds to be dead. And if they're really super dormant, and there are some seeds that have really very difficult dormancies that people are just having a great deal of difficulty trying to germinate them or learn how to germinate them, that can be another problem as well. But seed banking is the most efficient and effective method for storing plant material. It saves a lot of space, and it can save a lot of time. If you think about holding a sample in the freezer of 1,000 seeds, you might have a bag like this. depending on the size of the seed, or if the seeds are very small, it could be a very small container. But compare that to trying to maintain 1,000 plants and keep them going. The space you would need for those plants, the horticultural expertise you need to keep them alive, the resources of water and weeding and nourishment, those kinds of things, it's very expensive. And so if you can store that much material in a small space, it's very efficient. Well, seed banks don't just store agricultural crop species. That's really, really important. But it's also really important that we store a lot of material that is out there in the wild and back that up in case it becomes lost in the wild or extinct. And with the prediction that 40% of our plant species out there are threatened with extinction, seed banking becomes a really important endeavor. And I'll just give you a couple examples. This is the Millennium Seed Bank at the Royal Botanic Gardens in Kew and Wakehurst Place in England. Has anybody ever been there? It is a beautiful place. You may have heard of Kew Gardens in London. That is just outside of London, and it's a wonderful place to be. Wakehurst Place is sort of a satellite garden they have, which is about 30 miles south of London. It's very close to Gatwick Airport, so if you're ever flying into Gatwick and want to visit, it's just a short taxi ride away. But the Millennium Seed Bank was started in the early 2000s with money from the British lottery. I'm not exactly sure of who decided to do all of this, but they had money from the lottery that they decided they would put toward millennium projects. And you may have heard of the Eden Project in England. That was set up with that money, and the Millennium Seed Bank was a project that came out of that as well. And it has grown over the years, and the goal there that they've had is to bank all of the seeds that they can from all over the world that are seeds that can actually be banked. And we'll talk in a minute that there are seeds that can't be banked. But they have, I think, gotten to about, well, they said they have about 40,000 species at the moment. So they've certainly got a big chunk of the species that are out there, and 2.4 billion seeds with those 40,000 species. So a lot. And again, picture of a guy with his coat on in the seed bank. When it's minus 20, you want to keep warm. But they have beautiful grounds, and Wakehurst Place, they have gardens and things around there too, so it's a nice place to visit. There are other seed banks around the world that are really focusing on wild species. Australian Plant Bank, and then the Germplasm Bank of Wild Species in China are two of the largest, focusing on wild species, but there are a number of other ones around the world that are either have been developed or are being developed as we speak. And I can't leave seed banking without mentioning the seed bank that seems to be the best known around, and that's the Svalbard Global Seed Vault. It has gotten a lot of press in the news, and it's an interesting place. They built it in Norway into the earth with the idea that if it was below the permafrost, that it could stay cool enough to keep the seeds cool enough. They actually do have assisted cooling in it, but they don't have to cool it as much as they would if it were in a warmer place. So they have 1.1 million accessions of seeds, species from around the world. And what they do is they're really duplicating accessions that other places have. So they want to serve as a backup. And that's really a great idea, because if something happened to the Millennium Seed Bank, if they only have one accession there of something and it's the only one, then it's gone. But I know Millennium Seed Bank also has backups in other places as well. But the Svalbard Global Seed Bank is what they call a black box seed bank. That is, they take the samples, put them away in the seed bank, and they don't really do anything. They're not monitored for viability the way I mentioned at the USDA and Millennium Seed Bank and other banks are doing. So it's an interesting sort of backup for some other seed banks around the world. Well, we'll come a little bit closer to home here and talk about CREW, where I work, and the kind of work that we're doing. We are the research program at the Cincinnati Zoo and Botanical Garden, and the animal division of CREW was established in 1981 by Dr. Betsy Dresser. And the plant research division was established in 1988, which was just the year after the zoo actually became a botanical garden. And so they wanted to add a plant research program to CREW. And I don't know, has anybody been to CREW here? Okay, great. Yeah, we do have a public exhibit that you can visit in the summer. And it's just been redone, so maybe next year, if you're over that way, you can come by, and tours and that sort of thing come through as well. And just to orient you to the zoo, the circle on the lower left of that map is the Vine Street entrance, and then we're at the other end, that little square at the very tip over there. So we're sort of off the beaten path. But with the new elephant exhibit, which is going to be opened sort of just beyond us, we'll be a little bit more where people are around. Well, in the plant research division, we have some real particular areas of focus. And we focus, one of the areas is tissue culture propagation. And tissue culture is sometimes called in vitro propagation because it's done in glass test tubes or petri dishes and that sort of thing. The picture there shows one of our endangered plants growing in a test tube. And it's growing on a nutrient medium, which is like a gel. That it's sometimes we use agar, which you're probably familiar with. That particular plant is growing on a different kind of gel. It's similar to agar, but it's very clear, and so we can sort of see if there's contamination or if roots are growing. It's a little bit easier to see things going on in the culture. But sometimes we use agar because certain plants like that better than this gel that we use here. Everything has to be kept sterile. And so when we initiate a culture, we have to, if we take tissue from a plant that's growing outdoors, we have to sterilize it. And what we do there is surface sterilize it usually in a dilute solution of bleach. For a certain length of time, you want to kill all the bacteria and fungi on it, but you don't want to kill the plant, so you time it and rinse off the bleach and then put it in culture. And sometimes that works really well. Sometimes there's bacteria and fungi inside the tissue that can then grow out. So we sometimes add fungicides or antibiotics to the medium to try to stop that right at the beginning. But it is possible to initiate cultures from a lot of different species, and we've done that. The nutrient medium contains the kind of nutrients that you'd find in a fertilizing solution. If you're a gardener and use fertilizer, it's pretty much the same ingredients, just in different proportions sometimes. There's also sugar in the medium. And we add plant hormones to the medium. And these are natural compounds or synthetic copies of the natural hormones that help direct the plant's growth. So there are some hormones that help direct shoot growth and promote shoot growth. There are some hormones that help promote root growth. And if you're a gardener or have ever made cuttings from plants, you may have used what they call like root tone or some of these rooting compounds that they sell in the nurseries. And that's a plant hormone that will help stimulate the initiation of roots on the shoot. So we use those sometimes if we want our shoots to root. But this culture itself is a shoot culture, and we can just keep it going pretty much indefinitely by putting a shoot in there. It grows up and looks something like what you see on the screen. And you can see there's a couple different branches in there. And so when the nutrients get used up in the medium, we can go in and cut each one of those shoots, put each one into a new tube, and they will grow. And the hormones we put in there will help stimulate the lateral buds to grow out, and we get more shoots. And you can just keep doing that. And then if you want to root those shoots, you can take the shoot and put it into a medium that has a rooting hormone and get it to root. And then when you have a root and a shoot, you've got your whole plant back, and you can then take it out and put it into soil. And that is called acclimatization, when you remove it from the test tube and put it in soil. And these are just pictures. That's a picture of a shoot that now has roots on it, and then one that went into soil. And the purpose for these tissue cultures that we initiate and grow at CREW is to propagate these endangered plants. You can keep maintaining them in these in vitro collections as a bank, in a sense, to back up collections in the wild. But mostly we try to produce plants for restoration, and I'll show you a picture in a minute on that. And then we also provide tissues to cryopreserve, because if we have these cultures growing, and every couple months we have to go in and reculture them, it takes a lot of labor. And so if we can take those tissues and freeze them and put them in a bank that way, we don't have to worry about them for a long time. So this is just a diagram of the process. We take the plant shoot tip from the wild, put it into your medium, get it to grow, get roots, you get the shoots and the plants back. So that is the basic process for any of the species that we deal with. We have produced a number of species for restoration projects. This is a picture of people planting out our autumn buttercups. And we've produced about, over 1,000 of these plants for a restoration project that is in Utah. The autumn buttercup was a species that just grew in only one place in Utah. And so we were asked to help to try to propagate plants for that to bulk up the population. So over a number of years, we supplied plants for several outplantings. And we've also produced plants for South Florida, several species of rare ferns. There's the Cumberland sandwort, which we established a population of that rare species in southern Kentucky in the Daniel Boone National Forest. Avon Park harebells, Rugel's pawpaw are two other Florida species that we've sent back to Florida for restoration. And then the running buffalo clover, and also the new Kentucky clover. We're working on to provide plants for restoration. Running buffalo clover, you may have heard about that one, is a rare species, or it was threatened, federally threatened. It was just removed from the endangered species list. It was delisted just this year. And so that one, there have been enough restorations and new populations found that they felt it wasn't as threatened anymore. Kentucky clover, however, is one that is found in only two counties. It was just recently found as a new species in about two counties in Kentucky. And so we're now trying to work with that clover and propagate that. Well, when we deal with all of these tissues, our goal with many of them is to bank them. And we do that in cryopreservation rather than in a seed bank, because the plants we deal with are called exceptional plants. They're plants that really can't be stored in a seed bank. Let's see. I think I may have had my slides mixed up, but I'll just talk about exceptional plants here in a minute, and you'll have another picture of it in a second. But exceptional plants are the ones that either there aren't enough seeds to collect. Maybe the plant is not making any seeds, and so you can't seed bank that plant if there's nothing to bank. The plants may be sensitive, the seeds may be sensitive to drying, and that's another reason you can't bank certain species. Or they may be sensitive to freezing, and they can't be banked. Or they might be so dormant you can't germinate them afterwards. So what we do is we focus on plants where we need to bank them in other ways. And the way that we specialize in are with species that we need to start a tissue culture line from them and then bank them in liquid nitrogen. And let me see if I can, yeah, I'll just go to that slide because that reminds you of the 4 steps of seed banking. And exceptional plants can't be preserved in seed banks because of a roadblock at one of these 4 steps. So they need some form of cryopreservation to be preserved. Well, I mentioned before that water is the enemy of preservation, either in a regular freezing or in a in cryopreservation. Liquid nitrogen is what we are storing the tissues in when they are cryopreserved, and this the temperature of that is minus 196 degrees centigrade, or 320 degrees Fahrenheit, extremely cold. And at those temperatures, the biological systems of the tissues slow down, and you can maintain the tissues over long periods of time if you can keep them alive during the freezing process. And that is the key, because if you have tissues that don't normally dry out the way seeds do, and they don't have that desiccation tolerance, you have to do something different to them. And what we do is chemically cryoprotect them. And we work with a variety of species and put them in our frozen garden of Crew's CryoBioBank. And this is just a list of some of the things we have. We have some seeds, spores, embryos, and shoot tips. Well, I mentioned that we use a chemical cryoprotectants for our species, for our tissues that we cryopreserve. And the chemical drying that we use is done with a solution called PVS2, or plant vitrification solution number 2. And it's called vitrification solution because what it does, it removes enough water from the tissues and stabilizes the tissues enough that if you freeze it very, very fast, you wind up with a glass instead of crystals in your tissue. So instead of having those crystals form and break your cell, you just have a glass that is the same size and shape as the liquid that was in there, the solution that was in there. So the chemicals that are used for that are, it's a sort of like a chemical soup of ethylene glycol, glycerol, DMSO, and sugar. And those will pull water out of the cells. Some of them also go into the cells and stabilize the tissues and allow the tissues to form this glass, or that we say it's vitrified during the freezing process. Well, how well does this actually work? We have a number of species in our collections of different types of tissues. And a few years ago, we had a project where we took them out of storage, took samples out of storage of all of these kinds of tissues, and tried to grow them again and to see if they would grow. And we found out that for the most part, the tissues were preserved really, really well. And that was very exciting. They had been in there for anywhere from 5 to 20 years. And this included seeds. And now you may say, well, why are you putting seeds in cryopreservation? Because you can put them in a seed bank. Well, as I mentioned, some seeds don't go through the freezing process really well in a normal seed bank. But some of those seeds will last a little while, but they lose viability really quickly. And we call them short-lived seeds. If your seeds will only last 3 or 4 years in the seed bank, it's a lot of work to take them out, regenerate them, put them back, or go collect them again. If you can go to collect them again, because the seeds may, if they're from an endangered plant, you may have lost the species in the wild in the meantime, and you may not be able to get them back. So anyway, there are reasons to actually cryopreserve seeds because the seeds may be short-lived. And cryopreservation, storage in liquid nitrogen will keep them for much longer. We did pollen of the American chestnut. There's black walnut, apple buds, variety of species that lasted many, many years. Let's see. And just to finish up, I'll talk a little bit about oaks, because that is a group that we are focusing on particularly in our work at CREW. There are 500 species of oaks worldwide. About a quarter of those are threatened. There's 91 species of oaks in the US, and about a third of those are threatened. And oaks are of interest to us because their seeds are sensitive to drying, so you cannot put acorns in a seed bank. So we are cryopreserving oak tissues, and this just illustrates the process that I've talked about. You collect the tissues, get them growing in tissue culture, then take the tiny tips and put them in liquid nitrogen, and when you want to take them out, you take out those little tips, put them back onto a medium, and reestablish the culture, get shoots, get the roots on the shoots eventually, and then you can get plants back. And so we've tried this with several oak species, and in a recent study we did, we looked at 4 species and their tolerance to cryopreservation. One was the live oak, Hinckley's oak, which is a critically endangered oak from Texas, Quercus suber, which is the cork oak, and then Quercus gambellii, which is another oak. And you can see from this graph, the black bars show how much survival there was in liquid nitrogen. And the Quercus virginiana is the first species there, had pretty good survival, about 50%, but the other ones were much lower. Now, we did get some survival from them, so that was encouraging from the 2 species. So what we learned from this was that there's a lot of species differences in the way that these plants are going to survive. So we are encouraged that we got a little bit of survival, but we need to improve the situation. And so we are currently, we actually got a recent grant to work on oak tissue culture and cryopreservation. So we're continuing this work. And I'll just finish up there. Hopefully I've presented a case to say that plant species, not just plants in general, but plant species are important in conservation. There's a lot of global efforts going on to try to preserve them. And at CREW, we're focusing on exceptional species with tissue culture and cryopreservation to try to preserve those as well. So I will thank you for your attention, and if you have any questions, I'm happy to try to answer them. Yeah, the question is, what kind of containers are these plants in, or the tissues in when we freeze them? Yes, they're in little cryo vials, which are about this big, and we actually, in the method we're using now, we actually freeze them directly on a little strip of aluminum foil. We put the foil into the liquid nitrogen, and it freezes them, and then we put the foil with the tips on a into the vial, and then put that in the CryoBioBank. Yeah, yeah, you have them in something. The other, yeah.
Valerie, back to some agricultural seed banks. While I understand that's all very important, with modern and industrial agriculture, in practice, it seems that we're eliminating diversity of species, whether it's corn or soybeans or bananas.
Mm-hmm.
So don't we need to have more diversity in actual practice, I guess is the question.
Yeah, the question is about the sort of the conflict, I guess you'd say, between trying to save all of these diverse races and varieties of crop species versus modern agriculture, which is trending toward growing monocultures of sometimes genetically modified organisms, or just crops that they've produced, hybrids and such like that. Yeah, I would agree that we need to be sure to not lose these varieties. I know there's a lot of breeding and a lot of work going on to try to describe the traits that are in these collections. That's a whole field in itself. Because people know that, well, I've collected some from here and some from here and some from here, but what do those lines actually have in them that could be useful? And so there's a lot of study going on in that. And I think the more we find out about that maybe those can be then integrated into the bigger agricultural system. Yeah.
And along that line, I do some volunteer work that interacts with a group in here in Cincinnati called the Cincinnati Permaculture Institute, which works on integrating different organic and perennial type plants to work together to grow. And it's interesting how there is a lot of work being done to try to use natural methods to grow agriculture. There's actually a lot of work being done to do that kind of thing, to not work on monocultures, but to have, there is that. And the Cincinnati Permaculture Institute is actually pretty well known in the United States
Mm-hmm.
for doing that kind of stuff. So that's really interesting. It kind of works along with what you're doing.
Right, right. The comments were about planting diversity, basically, of groups of species. Yeah.
And building off of that, I remember there was a concern years back about like a banana blight because all Chiquita bananas are genetically identical, and they realized that line was susceptible to a particular disease, and it was kind of like, well, if one's susceptible, they all are because they're genetically identical. So whereas when you have something more genetically diverse, even if it's the same species, there will be members of the species that have immunity when they're each genetically unique.
Mm-hmm. Right, right. That genetic diversity can really build in resilience to fight against things like that, environmental or disease and such. Just think of the potato famine in Ireland was the same thing, that they had a monoculture basically of potatoes that was susceptible. Other questions? Yes. Mm-hmm. The question is, what kind of plants do I grow at home? Things that can take a lot of stress tolerance, basically, because I don't have a lot of time to. I don't have a whole lot of house plants in the house, but I do like to plant things outside. but we have a really shaded area. So I have a lot of hostas, and then I discovered we have a lot of deer, and the deer love to eat hostas. So I'm trying to diversify with my own garden there. So, yeah.
My name is Madavi.
Hi.
All pronouns are fine with me. Thank you so much for your work, Valerie. I really appreciate you coming here. It is indelibly important work at this stage in the climate crisis. I'm curious about your thoughts on one particularly interesting to me dynamic of funding for your work, which there seem to be some contradictions from some of the donors to the Cincinnati Zoo, which include J.P. Morgan Chase and Procter and Gamble. Procter and Gamble specifically in Cincinnati responsible for the deforestation of the boreal forest, as well as the threatening and endangerment of caribou habitat there, the largest caribou habitat in the world. And it seems to put into relief the dynamic in which your work finds itself important in the climate crisis. It's very much like a preparation for doom, I would say, because unless P&G is being challenged in their deforestation practices, then they will just receive the clouds of, conservation, helping the zoo, helping educate people about plants, while at the same time being those responsible for the problems that you're attempting to address in your work.
Mm-hmm.
It seems like there's an unresolved contradiction there. May I ask, do you have any leverage in encouraging them to discontinue practices of deforestation and anti-environmentalist work?
That's a really interesting question, and I know other conservation organizations and botanical gardens have had that same question given to them. I know our particular funding for these kinds of research come from grants primarily. The zoo funding is sort of a different area than what I'm involved with, so I don't think I can really comment on that. It's certainly something that probably needs to be on the radar and thought about seriously. So I appreciate your comments on that. Thank you. Oh. Oh, thank you.
So much. All right, thank you, Valerie. Very interesting. And, this is just one of the myriad of issues that we have faced with our fragile planet. So hopefully with her work and the other side of Crew for wildlife that, hopefully they can make an impact to try to keep the planet viable. Just two quick comments. Next month, Wednesday, December 7th, we're going to be talking about fossils in our backyard. Those are going to be the fossils that you see about any rock that you pick up in Cincinnati. Those are fossils from the Ordovician period. Our speaker will be Brenda Hunda from the Cincinnati Museum Center. She's the curator of invertebrate paleontology, and she's going to be talking about all of those species, the trilobites and the bryozoans and the brachiopods. Cincinnati, 450 million years ago, was submerged in a very shallow tropical temperatured sea, and it resulted in a tremendous amount of fossil activity. And the Midwest, and Cincinnati in particular, is kind of the epicenter for these fossil remains. We're known globally as the place to come if you want to research the Ordovician area. So it'll be a really fun and interesting topic, and Brenda will probably give you a little hint about the upcoming exhibit at the Cincinnati Museum Center, which I think will come online sometime later in 2023. And then lastly, next Tuesday, it's Election Day, so if you haven't already, please vote. And with that, I say thank you very much. Thanks for coming out. We hope to see you next month.